स्टार्ट कर दूं मैं? अस्सलाम वालेकुम। Today we are discussing the adaptive immunity. So first, we will discuss the cells of adaptive immune response. These are the T cells and the B cells. These cells of the immune system, the adaptive immune systems. You should know the what is adaptive immunity. Adaptive immunity is basically that is adaptive from the environment or it is not a natural occurring immunity basically. So the cells involved in the adaptive immunity, these are the B cells and the T cells. Among the B cells, we have plasma cells that produces the antibodies. Among the T cells, we have cytotoxic cells, the helper cells. Helper cells are further divided into helper cell 1, helper cell 2, helper cell 17, and the regulatory cells. This picture is showing the maturation of the lymphocytes, B lymphocytes and T lymphocytes. So here you can see the bone marrow where the B lymphocytes and T lymphocytes are produced and they get matured. The T lymphocyte they get matured in the thymus. They further process within the thymus, and after the selection or the clonal selection, the uh, they are circulated or they come into the circulation. On the other hand, the B cells, they get matured within the bone marrow, and the B cells, they form the plasma cells, and they produce the antibodies. We will discuss in detail regarding the clonal selection of the T and the B lymphocytes. So, the specificity of the adaptive immune response, the clonal selection can explain many features of the immune response. What is clonal selection? Clonal selection is which clone should be or must be present in uh, a circulation or must come in a circulation after the processing within the thymus and the bone marrow. We are talking about both cells, B lymphocytes as well as T lymphocytes. So this clonal selection is important and it depends upon the specificity, it depends upon the signals required for activation and the discrimination between a cell and non-cell antigens. The T cell mediated immune response. The T cell mediated immune response is the arm of the adaptive immune response, which role is to combat infection, intracellular pathogens such as intracellular bacteria, mycobacteria, listeria, monocytogenes, viruses, protozoa, and etc. There are different types of the T cells. These are the regulatory cells and the effector cells. So regulatory cells are the helper cells, the CD4 cells, in the effector cells, which produce the effect, ultimate effect, that is the killing of the viruses, bacteria, and other pathogens. And these are CD8 cells. The functions of the CD4 lymphocytes, which are the helper cells, they activate the B cells. As the name indicates, they help other cells in immunity or causing immunity. So they activate the B cells, they activate the macrophages, they activate the cytotoxic cells. Now, the T cell receptors, here you can see the picture showing you the helper cells and the class recognition of the CD4 and CD4 cells. So the CD4 cells, the T cell receptors on a CD4, T cells recognize the pathogen peptide displayed on the surface of an antigen presenting cells. This antigen presenting cells, they recognize the CD4 cells. You can see that this is the T cell receptors and this is the antigen presenting cells. So this T cell receptor, they possess the CD4 and the antigen presenting cell they presents the antigen on the surface of this antigen presenting cells. So different types of helper T cells are produced depending on the type of the pathogen that is recognized. First, they recognize via the CD4 cells, they recognize via the MHC class 2, which is present in antigen presenting cells. They recognize each other and then they activate the helper cells. One, which activate macrophages and cytotoxic cells. The helper cell 2 cells help activate B cells, and helper cell 17 help recruit neutrophils and the macrophages. Now, what is the function of these CD8 cells? The CD8 cells are the effector cells and they perform cytotoxic function. They kill virus infected tumors and allograft cells. They kill vector mechanism. Either they release perforance, which is by cell membrane, or they induce the programmed cell death, apoptosis. Now, this picture is showing you the cytotoxic T cells binds to antigen on plasma membrane of target cells and directly destroy the cell. So this is the direct cytotoxicity of the cytotoxic cells. How do CD8 T cells kills an infected cells? This is the CD8 cells. Now they release the cytokines, they release the cytotoxic granules, and they mediate this mass interaction. So in this way, they are causing the direct killing of the pathogen. Different phases of the T cell response. The T cell activation is required to overcome the pathogens and produce the causing the immunity. So there are the three phases of this antigen in this activation: the antigen recognition phase, the activation and differentiation phase, and the effector phase. So first here you can see the antigen recognition. So the antigens, they are present on the T cells, the CD4 cells, and the CD8 cells. Now remember this, the antigen presenting cells either represents the MHC class 1 and MHC class 2. If this MHC class 2, then they mediate interaction with the CD4 cells, and after the recognition, and if it is mediate interaction with the CD8 cell, they mediate interaction with the MHC class 1. Now these, after the interaction, they activate, they causing the release of certain cytokines, and that causing the further clonal expansion. Now, clonal expansion, they increase in number of the cells, the T cells, and they are further differentiated into the effector cells, or the very important function of this adaptive or the activation of T cells is the generation of the memory cells. And finally, the activation of this leads to activation of the macrophages, the B cells, and the other cells. So this, which we have already discussed, the functions of the T cells. So this is the effector function of this CD4 cells. Now here you can see the CD8 cells when they interact with the MHC class 1, then they causing the release or release of the certain cytokines like the interleukin 2 and causing the clonal expansion. After this clonal expansion, this effector cells causing the killing of infected target cells and the macrophages activations and degeneration of the memory, which is a very important aspect of the this adaptive immune response. Now, first the antigen recognition, I told you something that induced the T cell activity, antigen presenting cells. The first signals, this is the T cell receptors, the antigen peptides, MHC double recognition, and CD4 MHC class 2 or CD8 MHC. I told you CD4 will interact with the MHC class 2 and this MHC class 1 CD8. The second signal, which is a co-stimulatory signal, there must be there the direction between the co-stimulatory molecules on the antigen presenting cell and corresponding receptors on the cell. These are the different receptors that represent all the T cells for the activation of the or they can act as a second signals. So this interaction here, you can see this interaction, the CD4 cells and the T cell receptors. So addition signals, you can see this, the receptors of the helper T cell lymphocytes, all these receptors, CD4, TCR, the CD3 and the CD28 and this. And this here, you can see the ligands of class 2 and its expression, right? So this is the AP antigen presenting cells. Here the MHC class 2 is presenting. So here there is a CD4 cells to mediate interaction. The second signal which is required for the interaction is CD28 and D7. So these interaction causing the further post-stimulation is required for the activation of the lymphocytes. And this last one, the causing the addition of which function is the addition, this LFA and the intracellular activation of the tube. So again, this is the same thing which you can see here, the antigen recognition and the T cell response. Here, if there is the resting co-stimulator deficiency, यहाँ पे co-stimulation नहीं हो रही है. If there is no co-stimulation, then there is the cells are in energy form. They are inactive form. they're inactive cells. While if there is an interaction with this P7 and CD28, there is a co-stimulation is present, and that leads to T cell proliferation and differentiation. So this is the normal response. APC expressing co-stimulator. Okay, there is interaction between the CD4 cells and the MHC class 2, and there is a co-stimulation. So that leads to the function of the effector cells. If there is energetic form, there is no co-stimulation, and then in recognition without co-stimulation. So that leads to no response. There is another interaction, which is the CTLA and this CTLA4 and B7. So this will further inhibit the further response of the, suppose if there is antigen recognition and there is T cell proliferation and differentiation, we need to stop it. ऐसा नहीं होगा कि ये हर वक्त ही कैरी ऑन रहा है और ये proliferation होती जाए. We need to stop this proliferation. For this, we need another binding, and this is among between these two molecules, which stop the further proliferation and differentiation of the T cells. So this is the recognition of antigen and the post-stimulated T cell express proteins that are involved in proliferation, differentiation, effector function of the cells. The biochemical pathway that link antigen recognition with T cell response consists of the activation of enzyme, recruitment of adaptive proteins, and production of active transcription factors. The proliferation differentiation of the cells regulated by the CD4 T cells, regulated by the cytokines, and the CD8 cells. These are regulated by the TH dependent. They are the basically this helper cell dependent or helper cell independent. That viruses infect the cells that highly express co-stimulation molecule and directly stimulate the CD8 cells. So in this picture, you can see the effect of the CD8 cells and the CD4 cells. This is a similar thing which we have already done. It's showing in the CD4 cells. Now here you can see this is the infected cells phagocytized by the host APC. That is, these are the macrophages and other WBCs. So represents the antigen after the breakdown, and they become the antigen presenting cells. So after the interaction, these helper cells causing the activation of the further activation of or causing the effector cells to produce their effect. Now, this CD8 and CD4 cell recognize antigen on APC that has ingested. This whole story is this. ठीक है proceed antigen presenting cell होगा. If there is an interaction between the CD8 and MHC, that produce its effect. If there is an interaction between CD4 and MHC class 2, then it produces its effect. So CD8 cells recognize antigen on infected cells, and there is a co-stimulation causing the clonal expansion and differentiation of the CD8 cells. Cytokines are required in T cell proliferation and differentiation. Activated T cells can produce cytokines, interleukin 2, 4, 7, 10, and express cytokine receptors that promotes T cells to proliferate and differentiate. Effector functions of effector T cells, CD4 cells. These are the helper cells that secrete the interferon. They express CD40, express fast ligand, kill fast ligand target cells, effect on lymphocytes. Interleukin 2 se hota hai, effect on neutrophils, is mediated by tissue necrotic factor alpha and beta. The helper cell 2 promotes B cell growth and Ig production, antibodies production, that mediates hypersensitivity reactions. The CD8 T cells, they cause the cytotoxicity, kills target cells, and produce necrosis and apoptosis. The necrosis, the perforance in granzyme, and apoptosis causing the granzymes and the fast ligand. These are the ways how they are causing the cytotoxicity. Characteristics of the CD8 T cell cytotoxicity to must be specificity, limits of recognition, and the high efficiency. This picture is showing the mechanism of killing of infected cells by the CD8 cytotoxic cells. So here you can see the antigen recognition and conjugate formation. Then there is activation of this cytotoxic T cells, and then they're causing the release of the granules, which I told you earlier about the perforins को करते हैं, granules को release करते हैं, and causing the apoptosis. So enzymes enter cells via receptors, and they causing the endocytosis and endocytoplasm via perforin-dependent mechanisms, causing the activation of the apoptotic. Now this antigen uptake or synthesis, the antigen presentation, and T cell effectors function. This is again the similar things which we have already discussed, the class 2 MHC associated presentation of intracellular antigen to helper T cells. This is the class 2 MHC, and in interaction होगी and reproducing the cytokines and activation of the lymphocytes, and then finally causing the effector function. Effector functions क्या है? Macrophage activation or the destruction of phagocytose, micro and B cell antibody secretion, antibody binding to microbes. Class 1 MHC associated presentation of systolic antigen to cytolytic T lymphocyte. So it is recognition by the class 1 MHC and the cytoplasmic microbody antigen presenting cells जो है वो आपके activate करेगा, class 1 की recognition करेगा, presentation होगी, and this CD8 cells interact with MHC class 1 and causing the activation of cytotoxic cells and causing the killing of antigen expressing infected cells. So this is two types of cell-mediated immunity. The cell-mediated immunity against intracellular microphagocytosis with ingested microbes, microbillion antigens and vesicles, and then we combine on and similar things, CD4 and the helper cells, and they causing the killing of these things. Similarly happening here, infected cells with microbes and cytoplasm and causing the killing of the infected cells. So there, these are the picture showing you the types of the immunity hypersensitivity, delayed type of hypersensitivity, and T cell mediated hypersensitivity reaction. So this delayed type hypersensitivity reaction is produced because of the CD4 cells, helper cells, and that causing the a delayed reaction, which we will learn in detail in hypersensitivity reactions. So this delayed type hypersensitivity reaction causing the inflammation and tissue injury, and the T cell mediated cytolysis, which causing the destruction of the cells or the lysis of the cells. So in this picture again, this is the similar thing which we have already discussed. This is a recap of everything. The B lymphocytes, the helper T lymphocyte, the cytolytic T lymphocytes, and natural killer cells. So B lymphocytes, antibodies produce करेंगे, neutralization of microphagocytosis and complement activation. The helper T lymphocytes, they causing the activation of the macrophages after the interaction with all these things produce inflammation, activation, proliferation, and differentiation of the NB lymphocytes. Then cytolytic T lymphocytes, they infected T cells expressing microbial antigens causing killing of the infected cells. Natural killer cells causing the killing of the infected cells. So this is the overall picture how these different B lymphocytes and the T lymphocyte works and against the pathogen and causing the inflammation and activation of other immune cells. Thank you.